Okay, so before we really dig into second messengers, let's talk a little bit about big picture stuff. Let's take a step back and see what are we doing? Okay. So you know that an action potential causes neurotransmitter release from a presynaptic neuron, right? So in the presynaptic neuron, neurotransmitter is released. That neurotransmitter crosses the synapse and binds, in this case, G protein coupled receptors. Now, once the ligand is bound, the neurotransmitter is bound to the G protein coupled receptor, a G protein becomes active. Okay, now these G proteins are able to then associate with the G protein coupled receptor. They're able to get rid of GDP, bind GTP, and now we think of them as active, right? Whether that's a heterotrimeric G protein, whether that's a monomeric G protein, these G proteins are now active, and they typically, what do? They target or activate effectors. What are effectors? We just talked about some of them, right? Potentially ion channels, potentially enzymes, and those effectors then go on to change or something in the cell to activate second messengers. And we're going to talk about examples of how effectors activate second messengers now. And when second messengers are active, right, they typically have other targets in the cell. So this goes on to target some part of the cell, some part of the neuron. So we've talked a lot last chapter about neurotransmitters. We talked before about G protein coupled receptors and G proteins, and we've just mentioned effectors, right? A couple of effectors. Now let's get into the second messenger business here. Let's talk about these. And we'll start by talking about maybe the most common second messenger. It's actually a simple one, right? We'll make a list of our second messengers. And so the one we start with is calcium. Calcium is kept in ultra-low concentrations within the cytoplasm. We're talking nanomolar concentration, so it's kept very low in a neuron. Now, compare this to potassium. Potassium is found in, I don't know what is 100 micromolar, right? That might be 1,000 times greater than the concentration of calcium. So calcium is extremely, extremely low. So any rise in this intracellular calcium causes some change. Okay. Now, once calcium enters the cell, it doesn't typically stay free. Typically, it binds to proteins. Some proteins are there to just buffer the amount of free calcium that exists, right? So there's proteins like calbindin. And all calbindin does is to sponge up free calcium, okay? When calcium really comes in, though, like when it floods in, right, it exceeds whatever calbindin can bind up, and it binds to other calcium-sensitive proteins. A common one is calmodulin. And so when calcium is associated with calmodulin, we'll write it like this. This is calcium complexed together with calmodulin. Now, calmodulin is a protein. Calcium is an ion, so you have this teeny tiny ion, or multiple of these teeny tiny ions, that are associating with a relatively large protein. Okay, and this calcium calmodulin complex is able to also have and go on to bind downstream targets. So other downstream targets, and we'll talk about them. All right. Where's the source of this calcium? Where does it come from? There are really two places. The first is the external environment. Okay. So whether you've got a voltage-gated channel or a ligand-gated channel, either one is capable of letting calcium in. That's shown in this red line here. And so now free calcium is present. The second way that we can get calcium concentration to go up is when we release it from intracellular stores. IC, intracellular stores. Okay, so places where calcium is kept quite high is in the ER, in the endoplasmic reticulum. Okay, and again, we're going to talk more specifically about the mechanism, but you can see that this thing here, what's called an IP3 receptor, or this ryanodine receptor, they allow calcium out. And so whether it's from the external environment here, or whether it's from these intracellular stores, right, and a gate on the endoplasmic reticulum being open so calcium can leave, the effect here is the same, right? We're increasing the amount of calcium that's intracellular. And what does that do? It binds up these effector proteins, okay? So, calmodulin, our example, we have a complex now. We have calcium calmodulin together. How is calcium cleared? Right, that's the other thing we should talk about before we move on. That says clearance. And so some ways that calcium gets cleared. Well, I mentioned already, right, that there are some proteins that just buffer the calcium, that don't take part in any more active activations, or they're not going to work in any cell signaling pathways. So you've got buffer proteins here, right? Just sponges, just sponging up free calcium. There are pumps that are moving calcium not in but out, right? So you've got a calcium pump or a calcium exchanger here. Right? And so we're actually actively moving calcium out. So we can say it's pumped out. Or it's put back in to these intracellular stores. Right? So you can see that there's this other pump here on the endoplasmic reticulum's membrane. And calcium is being driven back up into there. So it's, I think the term that I like is sequestered back. Right? Like a jury is sequestered, it's put away. Calcium's sequestered back into organelles like the ER. The inner portion of the ER has a lot of calcium. Okay, so again, we'll come back to the targets of calcium, especially when it's complexed with calmodulin, in a minute. Other second messengers we need to mention. I told you that oftentimes second messengers are enzymes. Or I'm sorry, that effectors are enzymes and they activate second messengers, right? So let's talk about the second messenger of cyclic nucleotides. Cyclic AMP and cyclic GMP. Okay, these are derivatives of ATP and GTP, respectively, and they're converted, right? So ATP, for example, is made into cyclic AMP by the enzymes that we were just talking about, by these effectors. Okay. So in order to convert ATP to cyclic AMP, this requires the activity of adenylyl cyclase. Right? This is an enzyme. So put this together. Activation of a G protein coupled receptor leads to the turning on and activity of the G protein. The G protein goes and activates adenylyl cyclase, and now this thing is on. And adenylyl cyclase is able to take free ATP and convert it into cyclic AMP. Okay? Cyclic GMP is similar. Okay? Let me see here. So again, there's an enzyme in the cell's membrane, guanylyl cyclase, that takes GTP and converts it to cyclic GMP. Okay? What happens now that we have cyclic AMP? Why is that a big deal? Because it can work through downstream second messenger targets, right? So cyclic AMP isn't the end of the pathway, and cyclic GMP typically isn't the end of the pathway. These things activate kinases. We'll talk a lot about kinases, right? And so you've got in the diagram over here, guanylyl cyclase or adenylyl cyclase, the enzymes stuck in the cell membrane. Right? And when these enzymes are on, they take ATP and GTP and convert them to cyclic AMP and cyclic GMP respectively. And now these molecules can go off and have other effects, right? Typically activating kinases. Not all the time. Another effect of these cyclic molecules, these cyclic nucleotides, is that they can bind ligand channels directly. So activate kinases or interact directly with ion channels. Okay? And so you see that this channel is cyclic nucleotide gated, right? So let's look at the one that's gated by cyclic GMP. Whoop. This won't pass any ions unless cGMP is associated with it, right? And so once cGMP binds it, now it's permeable. This is a tremendously important pathway in phototransduction. Okay. One important note is that these molecules are regulated by phosphodiesterases. So phosphodiesterases degrade cyclic molecules. So we need to have a mechanism to turn them off. Okay. Enzymes that break down cyclic molecules, cyclic nucleotides. So before we wrap this one up, let's just talk about a potential example pathway that we could possibly see, right? We might have let's draw a cell membrane with the G protein coupled receptor in it. Okay, so this is my G protein coupled receptor. And a ligand can bind this G protein coupled receptor, and when that happens, the G protein, right, which is made up, let's say it's a heterotrimeric G protein, alpha, beta, gamma, associates with the receptor. Okay, and what happens is the molecule that's typically present here, GDP, gets booted out, right, and GTP comes in. And so now the G protein is active and it dissociates, right? Alpha subunit and beta gamma subunit go do their thing. Alpha subunit tends to target an enzyme that's embedded in the cell's membrane, adenylyl cyclase. Okay, and adenylyl cyclase is off until the alpha subunit turns on this enzyme. And this enzyme, now being on, can catalyze the reaction that takes ATP and converts it to cyclic AMP. Okay, that's as far as we're at with this pathway. Okay, and we will pick it up here in the next video.